Thank you very much. Great. Okay. So thank you very much to the organizers, first of all, for inviting me to this event, Women in Data Science. It's always a pleasure to connect with others in data science and women in data science in particular, for sure. And today I'm going to talk to you about natural language processing of biomedical text. And in one of the previous talks, NLP was mentioned as not too, we don't have enough fancy figures. You will see something similar in this talk as well. But I will try to keep you entertained, I hope, with opportunities and challenges. So the agenda, a very quick agenda, I will be covering 3 different examples of how we use NLP in life sciences or in healthcare. First, I will talk to you about a very common use case or a product that we have at Roche, which is a healthcare professional. Healthcare professional means doctor, nurse, or any kind of caregiver feedback classification. So this is a simple, let's say, use case or product that we have, and then I will layer on the science as I talk to you about some of these challenges, especially related to data scarcity and imbalance in data, talking about scientific literature classification first, and then protein-compound interaction prediction. So trying to cover different aspects of the applications of NLP in healthcare and data science. So text data in the biomedical domain, all of our, the words that we speak or the language that we speak or that we write in our conversations or in our communication, everything is human-coded, whether it's natural language or whether it's DNA sequences, protein sequences, chemical formulae, right? And they can be represented as in string form, in text-based representation, in this human codified way. And this is what I will try to cover today, talking to you both about natural languages and how we process them in some of these use cases or examples, but also protein sequences and chemical formulae. So what I will also drive or try to mention is the fact that we know that artificial intelligence and machine learning is really unlocking these insights that we cannot extract from text-based data with the advances in large language models, etc. But the scarcity of annotated and balanced data sets challenges the use of supervised learning. And the data scarcity bottleneck then presents also an opportunity or a challenge and an opportunity to overcome to improve the efficiency of these machine learning approaches, and I will also talk to you about that today. Okay, so let's start with this simple, not so simple, but let's say straightforward use case, which is healthcare professional feedback classification that we have. The healthcare professional, as you can imagine, Roche builds these relationships with doctors or healthcare professionals and receives feedback from them. And then this feedback somehow needs to be triaged to the correct department, whether it's related to, I need more education on this drug, so it gets sent to the education department, or maybe it's an adverse event report, it gets sent to the correct department, etc. And this feedback needs to be processed as it is received. So here we have Lisa, because this was a use case that started in Italy, actually. And Lisa manually checks all healthcare professional feedback. Each month, she splits the free text into several sentences based on her experience, subject matter expertise. Then she classifies each sentence as insight and non-insight. And then the insight sentences are then classified into 7 areas according to the Roche department that will handle the insight. So here in the simple first step, we have, let's say, 7 classes, but we have also received requests to classify into many more, like hundreds of classes, to understand, to extract more information about the healthcare professional. The problem here is that it requires manual effort, it's time-consuming, there's usually an external agency that's associated, and inconsistencies in classification. Maybe you remember this blue dress that we had a few years ago. So we built in this healthcare professional feedback classification pipeline. We start with the data. We developed different connectors for data ingestion. We trained machine learning models. We predicted classes, and then we report and visualize. I mean, bread and butter type of work, right? So quite straightforward. But one challenge remains. Remember, I showed you the blue and the other color dress. I don't know who saw white in that. But one challenge remains where it's hard to annotate this data consistently. On the left-hand side, you see pictures of cats. On the right-hand side, you see pictures of not cats. But what about the ones in the middle? You probably need subject matter expertise to be able to identify which is a cat, which is not a cat, right? So you probably need some sort of subject matter expert rules to say, when you see a cat with as a mug, classify that as not cat, blah, something like this, right? Some rule. So we included the subject matter expertise in this loop and included an active learning in this pipeline such that the subject matter expert, and you can imagine that Roche has several of these subject matter experts, if not several hundreds and thousands, as I mentioned, like Lisa, to correct the mislabelings and to add labels to unlabeled feedback. So this is our first example of where supervised learning, a simple supervised learning pipeline, was not enough, and we needed to include active learning or human-in-the-loop kind of learning. And this healthcare professional feedback classification use case is powered by our internal NLP capabilities, which we call an NLP engine, where we use state-of-the-art machine learning approaches, continuously improved by including human in the loop with this active learning. It's scalable because it's internal, as I mentioned, to different labels and amounts of data. It's multi-language. You can also imagine that healthcare professionals, we have healthcare professionals or interactions with them, engagements with them in all of the countries. So this also needs to be multilingual. And it's fast and it saves money. So this is a very simple classification use case, where, as I mentioned to you, the opportunities of natural language processing with these really large language models and language modeling are really strong and powerful, but still we needed an extra step. Now we'll start to include some more science into this. The text data in the biomedical domain, you know as well as I do that these domain-specific languages are very highly specific to their context, right? So that's a challenge. This is known, but there are also language models that address that problem. But the other challenge is that this is also a problem of finding needles in a haystack. So, for example, one simple example is when you read a scientific paper, the information that you want to extract is maybe in one of those sentences where you want to extract, for example, the relationship between a protein and a compound, right? So it's you don't have to read the whole thing, except you often have to do it to find that information, and that becomes a needle in a haystack problem. And especially if you have multi-label text classification, this becomes doubly challenging because of class imbalance and co-occurrence. What is class imbalance? For some labels, there will not be enough instances of the data. And some labels will be co-occurring with some common labels. Let's examine this simple text where it's talking about COVID-19, it's talking about human, it's talking about ICU. So you can imagine human, for example, as a label is going to be in many different scientific texts. And COVID-19, with the explosion of that research field, is also going to be very common. But ICU is not going to be very common, right? So some of these labels are head labels, some of them are rare labels. And finding those rare labels, as I mentioned, is doubly challenging, especially when you have multi-label text classification where you need to assign several labels to this particular text. So here it's going to be human and COVID-19 and ICU, but then how will you also know to differentiate between human and COVID-19 and ICU, right? So that's called co-occurrence. And this is actually not a problem only in NLP, but it's also a problem in computer vision. And you will see that we have borrowed some ideas from the computer vision field for this. So one approach that we can use to address this problem of multi-label text classification is by loss function manipulation. So what is most often done is binary cross entropy is most often used. It's the simplest, or let's say the most straightforward or most commonly used loss function, but it's vulnerable to label imbalance due to dominance of head labels or negative instances. Resampling, reweighting, also used quite frequently, but when there's label dependency, meaning there's co-occurrence, this becomes a challenge. So we proposed a novel approach where it's basically 3 loss functions layered into one that we call distribution balance loss. As I mentioned, this has been also used in the computer vision field, but it's our application to this field, where 3 layers, focal loss, rebalance weighting, and negative tolerant regularization are used in this loss function to address this class imbalance and co-occurrence problem. And what we saw was, even for tail labels, distribution balance loss improved classification. So let's take a look at this one, this slide. So here, yep. This is your, let's say, the data set comes from PubMed. This is your total, let's say, success metric, where our success metric for this use case is micro F1. And as you see, support vector machine, binary cross entropy, and distribution balance loss, these 2 are basically our baseline models. And this is our micro F1 for the total, when you examine all of the labels. For the head labels, we do get some improvement. What does head label mean? Head labels means those instances where you have more than 50, those labels where you have more than 50 instances. And here's how the class imbalance looks like. So whenever you have more than 50, you see that there are lots and lots of examples of those. And so the micro F1 score is somewhat similar to SVM, but when you look at the tail labels, where you have less than or fewer than 15 instances, our distribution balance loss function achieves higher success rate, or micro F1 score. So loss function manipulation presents to us an easy, let's say, pipeline, or an easy all-in-one kind of setting where we can use multi-label text classification for long-tailed class distribution. So that was natural languages, meaning the language English or scientific, or sorry, not just English, but spoken language or scientific spoken language. But let me also, I showed you this slide for a reason. Let me also talk to you a little bit about protein sequences and chemical formulae and how we use NLP to address some of these questions around protein-compound interaction prediction. So protein sequences, maybe you're familiar with the amino acid sequence. So this is a well-known representation of protein sequences where there are 20 different amino acids, and each is represented with a single letter. And a protein sequence can be represented with this 20-letter alphabet. And in fact, the protein sequence is what contains all of the information that will lead to the final protein structure, right? So this is also known. Maybe you've heard of AlphaFold. And chemical formulae can also be represented in string-based representation and, sorry, because this is PDF, it doesn't show the animation, but the chemical formula can be represented in this SMILES, or Simplified Molecular Input Line Entry System representation, where each of the atoms and the branches are represented in the string-based form. And here, again, the compound information is in the string form, and then you can revert back to the three-dimensional shape. Why am I always referring to the three-dimensional shape? Is because actually proteins and compounds interact with each other through this three-dimensional shape complementarity, right? So this is actually what we're after. But not all of the protein structures are known, and not all of the protein structures in complex with ligand representation is known. But there's a lot of data on how proteins and compounds interact with each other, right? So meaning experimental data. So testing protein A against compound 1, did it interact? Yes or no? At which affinity? I don't know, 50 nanomolar. And there are millions of these data. Okay. Having said that, though, even though there are millions of affinity, this is called protein-compound affinity, there are millions of these affinity measurements, you cannot sample all of protein-compound space. But we also know that proteins and compounds interact with each other through these physical-chemical relationships, right? Physical-chemical rules. And this is where we propose to use these causal ideas, which I hope will give me 50 more years of career, as Negar promised this morning. So these physical-chemical rules are actually defining how proteins and compounds interact with each other. But I cannot sample all of this conformational or this protein-compound interaction space, either because it's very hard to synthesize that protein or synthesize that compound, or maybe it's very hard to measure that protein-compound interaction. So I will come back to this in a minute, but there are, as you can imagine, there is a huge body of work on drug-target interaction affinity prediction, where either use the structure-based or the sequence-based, as we are using here, the text-based representation of proteins and compounds to predict the affinity score of the protein and compound interaction. And with this, that we published a few years ago, maybe last year, I think, with this approach and similar approaches, we do achieve high performance in comparison to benchmark and to state-of-the-art. That's great, right? So you can make predictions about the affinity, the strength with which a protein and compound are going to interact with each other. But predictions for novel biomolecules is a big challenge. What if in your training set you don't have that ligand? Ligand meaning compound, right, the chemical. Or what if in your training set you don't have that protein? So you never learned, or your machine learning algorithm never learned what that protein is going to do in a new setting, right? So this is a big challenge, and even though I showed you this state-of-the-art and how proud we are, blah, blah, well, actually it turns out prediction for novel molecules, biomolecules is a challenge, and warm is the setting in which the training set has seen both the compound and the protein. Whereas cold ligand, cold protein, or cold both represent the case in which the machine learning algorithm hasn't seen those cases. And if you examine this, you will see that predictions for novel biomolecules remains a challenge. So going back to that causal, or that idea that says to us, actually, it's the physical-chemical rules that are defining how proteins and compounds are going to interact with each other. Well, if you've sampled only a portion of that sampling space, or of that huge sampling space, then most probably, if these dataset biases are outstanding, weak learners are going to learn these patterns. Let me give you an example. So there are several millions, I don't know, thousands of cases where kinase interactions have been studied. Okay, kinase is a very heavily studied protein because of its relationship to cancer and many other disease states. Whereas some of these proteins are never going to be studied, either because they're impossible to synthesize, as I mentioned, or maybe they're related to some orphan diseases, and you haven't seen, not many people study it, right? So kinases, that kinase space is very heavily sampled. Well, if you have more of that data set, more of those types of data, then you will also start to observe some of these dataset biases. And if dataset biases are outstanding, the weak learners are going to learn these patterns and learn these spurious associations and return to you every time it sees a sulfur, let's say, it's going to say, oh, this is going to be a high interacting compound, but that doesn't make any sense, right? So probably. So these rules that the machine learning model is learning are actually spurious. So we used a two-stage approach that we call debiased DTA to improve the generalizability of this drug target affinity prediction, where we have a weak learner first. The weak learner is, I won't go into the details, the weak learner learns these surface patterns, easy to detect patterns, and then assigns an importance coefficient, meaning if that protein compound interaction is unique, then that has higher importance. And this importance coefficient is actually what the strong learner uses in the next step as it progressively trains to guide the strong learner training. And this debiasing approach improves prediction performance. I won't go into the details of the numbers, but it improves prediction performance both for the warm setting, but also for the cold setting as well. So the idea is based on, can I use these causal, or let's say a physical chemical, the underlying energy landscape idea to learn more about how a protein and a compound are going to interact with each other. And we showed that this debiasing improves prediction performance. So I tried to talk to you about three very different use cases where I mentioned to you that finding data, or finding, let's say, learning from this text-based data, but this is true for most biomedical data actually, is a needles in a haystack problem. Maybe if you're studying computer vision, for example, finding the region of that slide where there is information about cancer is going to be only a part of that, right? So this becomes a segmentation problem, etc. So it's a common problem in biomedical domain. And we recently published a paper that summarizes this, the methodologies that go beyond supervised learning to address this needles in a haystack problem, such as semi-supervision, active learning, which I talked to you about, data augmentation, which I also talked a little bit, transfer learning, self-supervision, and weak supervision. So we tried to summarize all of these in a publication recently, focusing a lot on the biomedical side of why we need some of these methodologies. So 3 takeaway messages. Data scarcity and imbalance are big challenges for deploying generalizable natural language processing solutions, not only for natural language processing. I think this can be generalized to other domains as well, but here I focused mostly on NLP. And approaches beyond supervised learning can unlock the potential of machine learning by helping find those needles in a haystack. And as I mentioned to you, so I am the head of data science at Roche, and I am part of the IT organization, right? So as part of the IT organization, we are actually there to serve these products. But what we also focus on, and what we try to focus on, is collaboration, open science, open source, so that we can keep up with these recent advances, but also so that we can employ some of these methodologies to these common requests coming from business. So here's the team that has been working with me at Roche. We are a very global team. Actually, this is not up to date. Now we're 25, but anyway, I just wanted to show you some of the names. And the NLP engine team, I would also like to thank them because they have been the ones that have been driving all of our NLP efforts. And the NLP engine, I've talked about this NLP engine at several different conferences, so feel free to check that out. And then I would also like to acknowledge my WiDS University collaborations. So I recently moved from academia to industry, from WiDS University in Istanbul to Roche here in Basel. And I would also like to acknowledge them for sure, because most of the work that I presented also was in collaboration with Arzucan Özgür, who is a professor of computer science at WiDS University. And I'd like to thank you for listening to me, and once again, the organizers for inviting me. Thanks very much.